donations of Dr. Jung's family, and to honor the Professor Jung, which is actually, he was actually the founding department head of the Department of Biology, okay, of the New Asia College, of course, many years ago. And this is actually the 5th year of the Jung's lecture in Life Sciences series, and it's actually one of the lecture series celebrating the 70th anniversary of New Asia College. Okay, congratulations to the happy birthday to the New Asia College as well. As you may know, Sir Richard got the Nobel Prize in Physiology and Medicine in 1993 for his discovery on split gene, which is actually now taught in all fundamental courses in biology. I still remember when I was an undergraduate quite some time ago in the 80s. Okay. I was actually so amazed. In the textbook, I still remember there's EM image, electron microscopy image, showing the hybridizations of a DNA and RNA with this loop structure, which is actually demonstrating the genomic organization of eukaryotic genes into introns and exons. And actually, this is actually an honor for us to have met Sir Richard in person to make this great discovery. And another research interest of Sir Richard is actually on DNA modifying enzymes, such as restriction endonucleases and DNA methylases. And since 1992, Sir Richard has been working as the research director at the New England Biolabs. For those who work in the laboratory, in any standard molecular biology laboratory in the school or around the world, you know this name, New England Biolab. For example, my student, I just told Sir Richard that we have been amazed with the new product. It's a Q5 mutagenesis kit, which sort of make another kit obsolete, okay, in my laboratory. And today he's going to tell us on his recent work on DNA methylases and the use of bioinformatics to discover new bacterial gene functions. And without further ado, let's give a big round of applause to welcome Sir Richard.
Okay, thank you. So, what I plan on doing today is to talk to you about some work that is ongoing in my lab. It's been going for quite a long time. And I'll give you a little bit of background of how I got into it, and then tell you about the stuff that we're doing right at the moment. In nature, mainly bacteria, lower eukaryotes, methylation post-replication is quite important. There are 3 kinds of methylation that we come across commonly. 5-methyl-C, this is a methylation that you find in humans, in more higher eukaryotes, but a lot in bacteria. M6-methyl-A is very common in bacteria, found in some lower eukaryotes, and there's recently reports that it's found in the human genome too, although it's a little bit iffy about just what is going on in the human genome. 4-methyl-C, in which this methyl group is on the 4th position of cytosine, is only found in bacteria at the moment. It's not been found anywhere else. Sorry. So, I'm interested in methyltransferases for a whole series of reasons. First reason is that they're part of restriction modification systems. So I've sort of made a career out of working on restriction modification systems. First got started in this in 1972, a long time ago. At the time, there were just 3 or 4 restriction enzymes known. One of the things I like about DNA methylases is if you give me a genome sequence, a bacterial genome sequence, using bioinformatics, I can find the methylase genes with high accuracy and very easily. We have excellent programs now that can do that. Several of them, in addition to working as part of restriction modification systems, clearly have regulatory activity. The DAM methylase of E. coli is a good example of that. They support various epigenetic phenomena. In Neisseria meningitidis, for instance, there is a DNA methylase that is variably expressed and will determine whether Neisseria can infect someone or not. There are also very good examples of proteins that recognize specific DNA sequences, and they do this in a rather interesting way. Shown here, this is the DNA helix coming down, this is the DNA methyltransferase, and this is the base that is going to be modified by the methyltransferase. What has happened is the enzyme flips the base 180 degrees right out of the helix. This we discovered in 1993, and we discovered it in what I call the nick of time. In 1993 was the year I won the Nobel Prize, and one of the things you have to do is you have to go to Stockholm, pick up a prize, but also give a lecture. And almost everybody talks about all of the fancy work they've done, following up on the discovery they made. Now, in my case, I discovered introns and exons and split genes, and I hadn't actually worked in that area for many, many years. But just before I had to go, we made this discovery, and so I used this as the basis of the lecture that I had to give. And you know, if you have to describe a discovery, always good to do it in Stockholm. There are interesting people who listen to what you have to say there. So it turns out all DNA methyltransferases do this, and almost every enzyme that does chemistry on bases in DNA uses this DNA flipping mechanism in order to allow the protein to get access to the base so it can do the chemistry. Now, the other reason that I rather like DNA methyltransferases is that when you start to look through genomes, you discover that there are a lot more DNA methylases than one might expect. Much more than is accounted for by the biology that we know about at the moment. And this is always a nice area to be working in when you see something, it's quite widespread, but no one knows what it's doing. And maybe there's a big discovery to be made there, and if there is, I kind of would like to make it. But if one of you made it too, that would be okay. So let me tell you a little bit about the restriction systems. There are basically three fundamental types of restriction systems. The type 1 systems, which were the very first one to be discovered back in 1978 in terms of being characterized. If you want to know what sequence they're recognizing, in this case this one recognizes GAG, a bunch of N's, and then GTCA, you have to know what this subunit, the S subunit, the specificity subunit, what it recognizes. Because it recognizes the DNA sequence and either forms a complex with the methylase, in which case it will methylate, or it forms a complex with the methylase and the restriction subunit, in which case it will restrict. It turns out for the longest time, working at DNA methylase recognition sequences was very difficult. It was one of these projects that it took so long typically to do it that when postdocs or students would come into the lab and I'd say, oh, why don't you determine the recognition sequence of this methylase? They'd be very excited and they'd go off and they'd say, this is going to be a great project. And then they'd talk to other people in the lab and around them, or they'd read the literature, and I would guarantee within a couple of days they'd be back and they'd say, well, you know, that was a good project, but could you give me something else? They didn't want to do it because it took a long time. The type 2 enzymes, these are the ones that you all know about. These are the ones that you use for genetic engineering, for characterizing constructs you've made, and so on. These systems have two separate enzymes. One of them is the restriction enzyme that cuts DNA when it's not methylated. The other is the methylase that methylates the DNA and protects the genome against the action of the restriction enzyme. These operate as two independent units. So the methylase works by itself and the restriction enzyme works by itself. And one question that we've had for a long, long time is whether or not the specificity of these two enzymes would be the same. So all we know is that in order to protect against NOT1, this sequence must be methylated. But if the sequence GGCC is methylated, that will protect too. And so when you start to think about it, there is no good reason, as far as we knew, why these two sequences should be the same, why they should recognize exactly the same sequence. I'll return to that towards the very end of the talk, because it is not completely clear, even today, why they should recognize the same sequence. Now in this case, getting the recognition sequence for the restriction enzyme is easy. You take a DNA of known sequence, cut it, get 10 fragments, and you look and see, you say, well, what sites can occur, 10 of them, that are this far apart to produce fragments of a given length. And typically, you only have to look at 1 or 2 DNAs, measure the size of the fragments, run a bioinformatics program, and you can predict what the recognition sequence should be. It's really very, very easy to do this. This is not difficult at all. Methylase has the same problem that you have above. It was not easy to determine methylase recognition sequences. And to start off with, for many years, we just assumed that the methylase was going to have the same sequence as the restriction enzyme, because this was the way it was being protected. Only recently have we come to find out whether or not that is true. Now, down here is a system that we call them type 3 systems. These were actually the second system ever to be discovered. And in these systems, the restriction enzyme and the methylase operate together. The methylase can work by itself, recognize the sequence and methylate it, but the restriction enzyme needs the methylase in order to cut, because it is the methylase that provides the specificity. And so again, this was a problem. If you wanted to determine the specificity, you had to get at the methylate. And that was always very difficult. But then along came Pacific Biosciences and the procedure for sequencing that they call smart sequencing. And as soon as this came along, everything changed. And the reason it changed is shown here. So smart sequencing, basically, you have a single-stranded piece of DNA, you copy it with a polymerase. It's actually a 529 polymerase that's been very heavily mutated. It copies the DNA, but it's set up in such a way that when it copies a methylated base, it takes longer to copy that base than an unmethylated base. And the system is set up so you can measure the time that it takes to add one base after another. When it comes to a methylated base in the template, there's a gap, something called the interpulse duration. And by measuring these, you can work out where the methyl groups are on the template. When you then look and sort of measure these distances, how long it took to add the next base, you get a signal that looks like this, and clearly here you took a longer time, and that says that that base must have been methylated. And what this means is that by just analyzing the interpulse duration, the kinetics of this particular reaction, you can work out the full methylation profile for a whole genome. And what this means is, for instance, let's say you've got an enzyme that is methylated GATC and methylates the A residue, then every time there is an A in the template, you get this characteristic signal produced. And that sounded very exciting to us because all of a sudden here was a way of determining the recognition sequences for DNA methylases. And so I immediately wrote to PacBio talked to the guy who was running the show over there, Jonas Korlach, and said, we would love to test this because we happen to have a large number of cloned methylase genes. So we could send to him DNA in which there was only one methylation present. We thought we knew what it was because of the specificity of the restriction enzyme. Let's see if PacBio, if this system, can find the correct recognition sequence. So we started off by taking a few cloned genes that we knew about. Here was the set that recognized GATC and either methylate the A or the C. If it methylates the C, it can be N4C or N5C. Here were the results that came out. With M6A, you get a very nice signal. With N4C, you get a reasonable signal, but you also get a few extra peaks. And with N5C, you get a very messy signal. And in general, we can find M6A with extreme accuracy. We can find N4C with pretty good accuracy. It's rare that any mistake is made. For M5C, sometimes you get a decent signal. Sometimes you don't, but you can't rely on it. So PacBio is not good for looking at 5-methyl-C, but it's good for N6A and N4C. And if you look here, what I've done is to draw circus plots, just showing what's going on. The inner strand, the outer strand, and you see where the sites are methylated. So we sent over to PacBio about 15, 20 cloned enzymes, and every time we got a nice clean answer, and we could work out what everything is. Now in the 3 cases I just showed you, we knew which base was methylated. And then we thought, well, let's send some things over where we don't know. We know the recognition sequence, but we don't know which base is methylated. First one was this one, a very simple one, recognized as AATT, but we didn't know which A was being methylated. You look at the PacBio result, and it's quite clear it's the inner A that is being recognized. In this case, GAC-GTC, we didn't know whether it was the A or one of the C's that was being methylated, and we didn't even know what kind of methylation. And here it turned out it was the A that was being methylated, got a very nice signal. So we published these results, but then we thought we'd like to go further. And so we started to look, these are all type 2 enzymes. We thought we would like to look at type 1 and type 3 systems. These were the ones where we didn't really know the answers ahead of time, because these were the ones that were very difficult to do. In the case of the type 2 enzymes, we always assumed the restriction enzyme would give us the specificity for the methylate. So we looked at Echo A1, which has this recognition sequence. BCEJ1 has this recognition sequence. And we got very nice, very clean results. Here's another one, a type 3 methylase from Bacillus cereus. We knew neither the recognition sequence nor the base methylated. When we look and do the Circox plots, you find if you look on the outer part of the sequence, which is one of the strands of the plasmid, you get a nice signal. And the signal occurs in the sequence CGAAG. And when you look at the base that's methylated, it's this second A residue. This was nice because we knew that type 3 enzymes have the unusual property of just methylating one strand of the DNA. They don't methylate both strands. And so this was a nice way of showing that. So this all looked very promising. And so we did a series of clones, and we started to look and see what was going on. And then we thought, well, maybe we don't need to clone the enzymes. Maybe we can look at the whole genome and get the recognition pattern for the various methylases. And we started with a few strains where we knew there were only a few restriction systems present. By bioinformatics, we knew what was in the genomes. Chromohalobacter selexigens was the first one that we did. It's got a type 1 system up here. It's got the characteristic 3 genes, methylase subunit, specificity subunit, restriction subunit. And then down here, there were 2 solitary methylase genes. When we look at the PacBio motifs that come out from this, you see here's one, RGATCY. This is a typical type 2 recognition sequence, and so it must correspond to one of these methylases down here. And down here is a split sequence that is absolutely characteristic of the type 1 enzymes. The type 1 enzymes recognize 3 or 4 bases here, a bunch of ends in between, and then 3 or 4 bases over here. So we knew this was the type 1 system, and this one must be one of these two. But we cloned out the genes just to be sure that we were getting everything correct. And sure enough, this corresponded to this, and this one actually corresponded to the one we call CSA1. This one gave no signal on the genome, but when it was cloned, then we see a signal. It specifies M6A followed by B. B is anything but A. So it can do A, C, AG, AT, but it doesn't do AA. When you look at this, we discovered it was on a prophage. Now, prophages are bacteriophages that have inserted themselves into the bacterial genome, and typically they don't express the methylase when they're in the genome. Typically, prophages turn off all the genes except for 1 or 2 that are monitoring when it's ready, when it's a good time for the phage to excise itself and start to make phage particles and go off and infect something else. They don't usually express the methylase gene, and that's why we didn't see it over here when we were looking that everything was being methylated. We did a total of 6 genomes shown here, and within these 6 genomes there were 27 methyltransferases altogether, and of those 13 were new specificities that had never been seen before. And in each case, to be sure that we were making the correct inferences as to what gene was recognizing what, we cloned out the genes and checked everything carefully. So that was very nice. So it was clear that this approach was working. We tested it in every way that we could, and we thought this was going to be really nice because we would be able to look at bacterial genomes that had been sequenced using PacBio sequencing, and then from this raw data, just calculate the methylation patterns. And so in this way, we could find out both the methylases that were active, what their recognition sequences were, and also get the complete genome sequence. One of the first ones that we did was this E. coli strain that caused major problems in Germany. It was present in a variety of, I think it was spinach that was being sold, or it was some green vegetable being sold. 40 people died as a result of getting this strain. We looked at the PacBio sequence and found all of these genes were present. A large number of methylase genes were present. We cloned each one out to check what it was recognizing. There are a number of things that we weren't expecting that happened as a result of this. The first one were these 2 genes, these 2 enzymes, both encoded on prophages, and they recognize just A residues. So they methylate all the A residues that are present, but they're on prophages, and so you don't see a signal in the genome. It was only when we cloned them out that you could see the signal, and I'll show you an example of that in a moment. Most of these others are regular ones. This one is actually also on a prophage, GATC. Here's the DAM gene of this particular E. coli, and so on, coming down. When we looked at all of these, we also saw something else that was a little odd, and that was that here was a system on a plasmid. It just recognized M6A. It had a small recognition sequence. It did not methylate completely, and I'll tell you a little more about that in a moment, because that turns out to be particularly interesting. The others were fairly straightforward, nothing special about them. But the other thing we found is that in regular E. coli strains, the strains that we grow in the lab, there are 3 genes, MCRBC, MCRA, and MRR. These are methyl-dependent restriction systems. These are systems that will cut incoming DNA if it's methylated. So let us say a phage had methylated its DNA to try to overcome a host restriction system. These enzymes are there ready to cut it up and restrict it anyway. But what's interesting is that these genes, which are present in all the strains we grow in the lab, were missing completely from this pathogenic strain. And it turns out that one of the, almost I would say, prerequisite for E. coli to become pathogenic is it needs to lose these 3 genes. And it turns out that Salmonella strains also have these genes in them, and they lose it when they become pathogenic. It looks as though this is a marker for a strain about to become pathogenic. And the Salmonella people are very interested in this because this could have some relevance for disease. Now let me just show you some data on the 2. This is M.echo G1. You can see every A residue on both strands of the DNA is methylated. This is the kind of print you get from this. This is the consensus sequence down here. Slight preference for C beforehand, but so slight that you don't really notice it. This is the single-strand specific gene on a plasmid, and here's what it does. This is, again, a Circos plot, and you can see that there is a lot of methylation on this strand and almost nothing on the other strand. That is, it is strand-specific to the plasmid. We found another example of this in a Burkholderia species. Again, all of the methylation on one strand, virtually nothing on the other. Now you might ask yourself, how is that possible? How is it possible for a methylase to know to methylate one strand and not the other? Well, the first thing that one thinks about, well, is perhaps it's associated with replication in some way. Let us say it is part of the replication apparatus, and as it is going around the plasmid, copying one strand, it is methylating it also. Why might that be attractive? I take you back to this. If you want to replicate DNA, you have to open up the helix. If you want to transcribe DNA, you have to open up the helix. It turns out that helicases typically use this mechanism to open up the DNA. There is good evidence now that transcription starts opening the DNA by base flipping. DNA replication too, there is some evidence that base flipping is what gets the starting point for opening up the helix. And so we thought, well, maybe what's going on is that this methylase is a part of the replication machinery, and we've now shown that it co-purifies with DNA polymerase 1, which is the replicative polymerase here. But the most telling experiment is shown here. If it is associated with DNA replication, then you might expect that if you changed the orientation of the origin, the alternative strand would get methylated. And that's exactly what happened. Sorry. Okay, if you look at the Circos plot here, when the origin is facing this way, it's the inner strand that gets methylated. When it's facing this way, it's the outer strand. Which looks very interesting. It looks as though this methylase is serving as a helicase within the replication complex, but why is it still methylating? Well, it could be that it doesn't need it, it's just an accident, it just has retained this activity, it isn't really important. But then there's another explanation that is possible. And that explanation is if you're a plasmid and you want to transfer from one strain to another, you would like to avoid restriction systems. One way you can do that is by methylating your DNA, and you think, well, maybe you could methylate all of it. But if you do that, as we can do with M.echo G1 or 2, then that has problems getting into strains, because when a strain is completely methylated, it interferes with replication, it interferes with other processes. And we know that in E. coli, if we take this plasmid and methylate it completely, it can't go in to E. coli very well. But if you just methylate it a little bit, maybe 10%, 15%, then it goes in just fine. And we think that that's maybe what's going on here. That just this partial methylation helps the plasmid get into the strain. It's part of the replication machinery. And here is something else. Methylases are very slow enzymes. They're not rapid enzymes like restriction enzymes, which cut everything in next to no time. Methylases are very slow. One of the things you would like to do when you're replicating plasmids is not to make too much DNA. Because if you make too many copies of the plasmid, they start recombining. And they clog the recombination machinery and they cause problems. And this has been well documented. And so maybe the reason that they use the methylase as the way of serving as a helicase is to slow down replication so that it doesn't go too fast. So here was something totally unexpected. It was good we cloned this gene, otherwise we would never have discovered this. But now if you look in GenBank, you will find there are thousands of plasmids that have this methylase gene. The methylase gene is well conserved. It's 227 amino acids long. Plus or minus one amino acid is in thousands of plasmids. This is just to give you some indication of how we've done in terms of discovering specificities for type 1 and type 3 methyltransferases. The first one was discovered in 1969, and you can see until 2012 there were only very few methyltransferases where the specificity had been determined. And because they occur and they're important for both type 1 and type 3, this meant the specificities of the restriction systems were unknown. But what's happened, thanks to PacBio, is the numbers are just shooting up. The dashed lines illustrate motifs that have been found but have not yet been assigned to genes. So for instance, you can imagine if you've got a genome and it's got two type 1 systems in it, and you get two patterns, how do you know which is which? Well, when I come back and talk to you next time, I can tell you exactly how we can do that, because we've been working on some algorithms to do that. We now can do that as of a short time ago. We've also, during the course of doing all of this, discovered some new ways of protecting DNA. So for type 3 systems, all of the ones that were known until relatively recently used M6 methyl A in order to protect. But now we have found quite a few use M4 methyl C to protect. Same thing with type 1 systems. We thought all type 1 systems used M6 methyl A. We have hundreds of examples, but now we found a few that use two different. They use M4C on this strand and M6A over here. And when these systems occur that use M6A and M4C, there are always two methylase genes. One gene is responsible for M4C, the other is responsible for M6A. And so that becomes a nice bioinformatics signature, tells us when we have these kinds of systems present. This is the specificity problem for type 1 systems and showing the architecture. There are two systems, two recognition sequences rather. And they're recognized by short sequences in the specificity subunit, 100 to 150 amino acids long. The one TRD1 recognizes the N-terminal sequence. The one over here recognizes the C-terminal sequence. And they are separated. Anywhere from 4 to 9 non-specific bases can separate these two. When you look at individual specificity subunits, most of them have just two specificities. Specificity TRDs, TRD1, TRD2, but there are some that have 3 and some that have 4, and we don't know how these work and how they interact with one another terribly well. when we look at these submotifs that are recognized by the TRDs, they can be very, very variable. They can recognize W, which is A or T. They can recognize S, which is C or G, and so on. A lot of variation. And the bottom line is, when you start to look at these and calculate how many theoretical possibilities there are, the numbers are enormous. To a point where when we look at a new genome, most of the time when we find a type 1 system, it's one we've not seen before. It's a new combination. These things are recombining very rapidly, and as you might expect, one of the problems that bacteria face is that for every bacterial cell, there are 10 phages. And so they're constantly under attack by phages. And in addition to restriction systems, the CRISPR systems that you all know about for editing DNA, these also are bacterial defense systems. And there are a lot of different bacterial defense systems. They spend a lot of time protecting themselves against phages. If they didn't, they wouldn't exist. So what have we learned so far? Well, first of all, there are many solitary methylases that don't have a restriction gene, or at least not one that we can recognize, either next to them, which is how most systems operate, or even at a distance, although we have found a couple of systems where the methylase gene, the restriction enzyme gene, is a long way away. One of the reasons they stick together is because if they transfer from one cell to another, you need the methylase to protect against the restriction enzyme, and so you need both genes. They're really a toxin-antitoxin kind of situation. And so they stick together, they hang around together. There's a lot more variability in recognition sequences than we ever expected, based upon the very early work that was done mainly in Noreen Murray's lab in Edinburgh, or Tom Bickle's lab in Basel. It seemed that there were not going to be that much specificity. They thought there was not going to be a lot of variation. In fact, they thought they could think about them in terms of just a few classes that they would all fall into. It's clear that's not true. The type 2 methylase specificity, referring back to my earlier point, they usually match the specificity of the restriction enzyme. And there's only one or two instances that we have where that is clearly not true. And in those cases, the methylases tend to recognize one variable base within the sequence of the recognition sequence of the restriction enzyme. So again, it's this situation with not one. If you imagine you methylate GGCC, you protect against every sequence that has that as the central tetranucleotide. In fact, if you had to make a general rule, it's that they're the same. And think about it, why should they be the same? I had never thought about this until I gave a televideo seminar to a group of Russians in Novosibirsk. I was sitting in a hotel room in Japan. I gave a video presentation to this group in Novosibirsk, and after it, one of the students, and I'm still trying to find out who it was, asked me, well, is it possible that what is being selected is not the restriction enzyme for protection, but the methylase? And all the restriction enzyme does is make sure that the methylase keeps honest in terms of the sequence that it's recognizing. And perhaps it's really the methylase that is being selected. And after all, the methylase had to come first. You can't evolve a restriction enzyme without a protected methylase. And so maybe these methylases were around for quite a time before the restriction enzymes evolved and made sure their sequences were preserved. That's rather interesting because it suggests that methylases, even when they're part of restriction enzymes, may be doing something different than we had imagined. They're not just there for protection, they actually have some other functions. We've also found that there are many, many non-specific methylases found on prophages. It's really quite remarkable how many phages and prophages have methylase genes on them. But when you think about it, well, it sort of makes sense. They want to overcome the restriction enzymes that they find in the host cells that they're trying to get into. So let me tell you some stories about common orphan methyltransferases. So GATC, this is found in the Enterobacter. It's in all of the Enterobacteriales pretty much have it. A lot of strains have it. It's a very common methylase. We know it's involved in mismatch repair, so it marks the parental strand against which repair should take place. It's also involved in DNA replication. The origin of E. coli has to be hemimethylated and then copied in order to determine when replication should begin. In the Colobacter, there is this methylase that recognizes GANTC. It is totally responsible for cell cycle control. And so this methylase is an important element for controlling when the cell cycle, which aspect of it should take place. In the Burkholderia, there's a methylase that recognizes GT, WW, W is A or T, AC. It's in all of the Burkholderias. It's in a number of Clostridia too. What is it doing? We don't know. There is no corresponding restriction enzyme. And unfortunately, the company where I work, we can't study this because all of these wretched Burkholderia are pathogens. And so I've been trying to find a lab working on Burkholderia that will look at this and see if we can work out what is the problem. What is this methylase doing? In the Clostridia, there is this interesting methylase. It recognizes CA5 and just methylates the last A residue. It is present in every strain of Clostridia. And we, over the course of the last year and a half, have been working with a group at Mount Sinai Hospital in New York, and they have come up with something very interesting. It turns out that if you knock out this methylase from a Clostridia, it can no longer sporulate. Now, if you think about it, that's pretty exciting, because one of the real problems that medics face is when people go to hospital, they often pick up Clostridial infections. In particular, Clostridium difficile is a very nasty bug, very difficult to get rid of. And one of the reasons it's difficult to get rid of is likely because it forms spores. So as soon as an organism forms spores, it becomes resistant to antibiotics, can't do anything about it. You wait, you stop the antibiotic, and then it grows up again. And so Clostridium difficile is a major problem. Maybe this is a great drug target. If we could knock out this methylase, that may be very helpful. Finally, on this topic, I just want to talk about this CAGM6AG methylase that is found in Salmonella. It is found in every Salmonella, but it's part of a type 3 restriction system. As far as we know, if it works as a restriction system, it doesn't work terribly well. Salmonellas typically have type 1 systems in them, which work really well, and it has other systems very often that work well. It seems unlikely that this is just there for protection because it's in every single species. It doesn't vary. So what's it doing? Well, maybe this idea that the methylase is what's being selected is good here. But again, unfortunately, Salmonella is a pathogen. We can't work on it at our company, otherwise the employees would go crazy. And so we're trying to find a Salmonella lab that will look at this particular property to see what happens if you knock this out. And does it affect pathogenicity? Does it affect transcription in interesting ways? What's it doing? What is it doing? Now, I can't finish this without telling you about the advantages of PacBio sequencing. PacBio sequencing has really revolutionized bacterial genome sequencing. First of all, I love it because the methylation specificity is easily discovered. So here, just for doing the sequence, you also find out something about gene function. You know which genes are active, you know what they're recognizing, and now we can usually, I would say in at least 50% of the cases, match the genes that are responsible for methylation. So here we get functional information just by doing the sequence. Second thing is, closing bacterial genomes is really simple. PacBio did very long reads. Now you can get up to 50 kb in a single read. And that makes it easy to then put the genome sequence together. You know, if you try to do this with Illumina, you've got 100 base pair, 150 base pair reads, and so the jigsaw of trying to put the whole thing together becomes very difficult, and usually you can't do it without doing some additional experiments. With PacBio, we have not recently come across a genome that we couldn't close the sequence from a single PacBio run just by doing a little computation. And so that's a big plus. And finally, PacBio is a very accurate method of sequencing. And the reason it's accurate is interesting. It's accurate because you read every base many times, 40, 50 times, while any given run is only maybe 85, 90% accurate. The errors are random. They're completely random. There is no systematic error that takes place. And so that means that after you've read it 30, 40, 50 times, you know every base very accurately. The same is not true for Illumina. Illumina has very specific errors that it makes every time. We're starting to learn what those are, but we don't know all of them yet. So Illumina has systematic errors. They're completely removed with PacBio. If you want to do very large genome sequences, then PacBio can be a little expensive, and PacBio at that point is best aided by also using Illumina to get the large amounts of data. So I'm a huge fan of PacBio, and I have to say I don't own any stock. I just look on. I love them because they've enabled science that was previously impossible to do. Now, one thing I told you about at the very start was that PacBio is good for N6A and N4C. It's not good for 5-methyl-C, but we've recently come up with a good way of looking at 5-methyl-C recognition sites. And that uses this enzyme, MSPJ1, which is an enzyme that recognizes 5-methyl-C something something R. So it needs 5-methyl-C, and then 2 bases beyond that it needs an R. And what it does, it cuts on both sides of this sequence. So if you imagine that you have a symmetric sequence here, you end up with fragments that are 32 base pairs long, approximately. Varies a little bit depending upon exactly where the 5-methyl-Cs are located next to one another. And if we take a typical bacterial DNA, this is what we get. You see here, this is 26 base pair, or actually 30 base pair in this case. So you get a big smear, but then you see a nice band down here. And this band contains the recognition sequence in it, and it's specifically positioned relative to the end. And so by sequencing a large number of these fragments, you can get the 5-methyl-C recognition sequence very accurately. We've done this on a large number of genomes now. Works really well, and we're about to write, well, we are in the process of writing this up. It will be published very soon. But it's a very nice complementary method to PacBio for looking at bacterial methylase sequences. We've also been looking to see if we can get the complementary enzyme to MSPJ1, something that will recognize a Y at this position, or better still even an N that just recognizes 5-methyl-C. We've not found it yet, but we're doing a metagenomic screen, and we actually have a very good assay that we can use to try and find the enzyme that we're missing. If we can do that, we will be able to do all 5-methyl-C recognition sequences for these kinds of methylases very easily. Just want to thank the various people at NEB, Ian Murray, Brian Anton, and so on, at Pacific Biosciences, Tyson Clark and Jonas Korlak, and then a large number of people. We've had collaborations now with 170 different labs who've sent me their PacBio data. We analyze it, send back the results. Some publish with us, some publish without. We don't mind. We're interested in the data because the more data we get, the better we can match recognition sequences with gene sequences. So that's been also very rewarding. It's really nice to collaborate and get to know people in many different fields, and it's quite surprising. Finally, I just want to make a plea. One of the things that I do in my spare time is to try to organize the Nobel laureates to help with a variety of causes. One of those is the pro-GMO movement. There's a website up there, supportprecisionagriculture.org. If you don't come to my third lecture, which I'm going to give specifically on the topic of GMOs, I would encourage you to take a look at that website. There's a lot of information there about why you should support GMOs, including links to references to lectures that I've given. There's just a huge amount of stuff there. Basically, everybody should be pro-GMO. The developing world really needs GMOs. They are safe. There is not a single example in the last 30 years where they've been anything but safe. Imagine we've fed billions of animals, cattle, sheep, billions, with GMOs, never a problem. People have been eating them. Papayas from Hawaii, half the US population has been eating them. Not a single problem. And this is just an issue. Greenpeace got it wrong. The anti-GMO people, they just made a mistake. They should just admit it and get on with doing all the good stuff they can do. So, thank you for listening. Happy to take questions.
Okay, take questions.
I'd like to ask, besides the sequencing, have you also targeted Oxford nanopore?
Yeah, so we do some Oxford nanopore sequencing. Okay, the question was about Oxford nanopore as an alternative way of getting very long sequences. So Oxford nanopore claim that they can detect methylated bases. We can't when we try it. Other groups have not been able to. They also have a problem that they're very inaccurate still. It's just not an accurate technique. For resequencing, it's quite good and it can be very helpful. But if you want to do de novo sequencing, then it really isn't that good because the accuracy is just not that great. But lots of people are using it for resequencing, which is fine.
Thank you very much. Because I had a little bit experience in trying the early stages of the nanopore, and I was also trying to take some experience, and this is very good advice. Thank you very much.
Well, the Joint Genome Institute that's part of DOE at Walnut Creek, they have been doing a huge amount of sequencing. They spent quite a lot of time trying the nanopore. They stopped it. They just stopped using it. They found it was wasting too much time. Yes.
Thank you for your excellent talk. So I have a small question about the physiological meaning of those sequence-specific methylation. For example, do they have any correlation with the gene expressions, patterns, etc.?
Yeah, so, there are a number of papers out there showing that yes, by changing the methylation patterns, you change gene expression. The example I mentioned earlier on, Neisseria meningitis, and there is a type 3 system that's variable, it switches on, switches off. In Helicobacter pylori, there are systems that have a sequence at the end that can be variable, and that can actually change the specificity of the methylase, and in that case that also has effects on transcription. So there are plenty of examples where this happens. We're not yet able to predict very well because we don't have enough experimental evidence looking at what the effects are. Okay.
There was one experiment to switch the origin of replication.
Yeah.
So the gene expression pattern changed?
We didn't look. We've not looked. We were interested in the fact that this methylase could be a part of the replication system, and so that was our focus. I don't know. If you buy my argument that this partial methylation helps the plasmid to establish itself in another cell, you don't really need to worry about transcription.
When I asked the question, I thought other people might want to hear too. Plasmid requires some ways to transfer from one cell, one bacterial cell to another. I mean, there's a species differences, and how would you replicate these plasmids or transfer these plasmids?
Right, well, typically there are transfer factors that will get everything started, and what they do is typically initiate replication and then perhaps do something to membranes to allow the plasmids to transfer. But the principal thing we know about is that they definitely tell this plasmid it's time to replicate and go off into interesting things.
Okay, so plasmids can transfer some kind of resistance.
Yeah.
Do you think this is really behind the multi-drug resistance?
I don't know. You know, one of the problems, almost nobody works on plasmids anymore. All of the interesting stuff, biology, biochemistry, was done back in the 70s, 60s, when people didn't have the same tools we have today. I personally do not want to become a plasmid biologist. I'm much more interested in methylation. So we hope that when we publish this work, that some people interested in plasmids and replication will follow through, and perhaps they'll be able to answer some of the questions you have.
Yeah.
You know, there's a limit to what one can do. And,
Right, and also
my focus is on methylation.
the interesting part is also the prophages.
Yes.
Play in this thing.
Yeah.
Yeah, I'm just thinking about
Yeah.
evolution, meaning all these things interacting with each other.
Yep.
Yeah, whether or not the world is still changing very rapidly in the microbiology.
We're still evolving, yes.
Yeah.
Evolution works.
You know, Bacteria, what's the bacteria? Bacteria? The bacterial.
Yeah, what about
the cystic fibrosis patients. So we want to collaborate with people working with cystic fibrosis.
Well, so I'm very interested in the microbiome and the way in which the microbiome influences health. We're starting a research institute in Shenzhen that will focus on the microbiome, and I'm hoping that some of the young people working there will make some progress in terms of using the products of the microbiome.
Easy?
Okay. So do the restriction enzymes evolve from a general nuclease or a non-specific nuclease in response to a presence of a particular methylase?
All we know about restriction enzymes is that they're evolving much more rapidly than methylases.
Yes.
So for instance, if you find a restriction system where you've got the sequence of the restriction enzyme, you've got the sequence of the methylase, you go into another bacterium and ask how well are these two conserved. The methylase is usually conserved reasonably well. The restriction enzyme, very often you can't even recognize it. If you've not done the biochemistry to know it was there, you wouldn't know it was there. And for a long time, the working definition of a type 2 restriction enzyme was that it was an open reading frame with no homologs in jet bank that sat next to a methylase. So they're evolving very rapidly. We don't know why they're evolving so rapidly, other than that selective pressure on them may not be so high.
But it must evolve from something else. So what's the starting point?
Well, it's actually very easy to make enzymes that will cut DNA endonucleases. 3 amino acids will form an endonuclease. So it doesn't take much to cut DNA chemically.
More questions? To the front.
Yes, Dr. Rhodes, I totally agree that the GMO is actually the answer for the hunger problem. But for developing world, of course, that's the most important thing. Let's say for developed countries, would there be more subtle and chronic problems with the GMO that we're using now? Say your favorite topic, the microbiome, would that be affected, and would that be a chronic and subtle problem?
Okay, well, I think to really get at that, there are two things to think about. One is traditional breeding mixes two genomes, and then you try to get rid of the genes you don't want, but you don't get rid of all of them. So any plant that has been traditionally bred has a lot of genes in it, no idea what they're doing. It's quite imprecise in terms of making a new plant. Is that something we should worry about? Well, we don't actually test it. We never test to find out if that's a problem. The GM controversy arose for the simple reason that Greenpeace found it a very good way of raising money. They have raised more money as a result of that than any other campaign they've ever had. Their annual budget in Europe is estimated to be between 400 and 500 million euros a year, and most of that money has come in because of their anti-GMO stance, because they scared the Europeans, and to no effect. There was just no evidence, zero evidence. You know, in 30 years of using GMOs, eating GMOs, there's not been a single credible problem. The antis are always finding a little bit something here and there. So Seralini's paper is the one you hear about all the time. He showed that, he purported to show that feeding rats that were already cancer prone gave them cancer. He was forced to retract that paper. The journal made him retract it. And then he goes and publishes it again in a non-peer reviewed journal. And now the anti-GMO people point to that paper instead. Right? They ignore the fact that scientific consensus said the thing was flawed. Last year, the European Union funded some people to repeat his studies and showed that there was no difference. Whether you were eating GMOs or whether you were eating regular food, there was just no difference with the incidence of cancer in these rats. And, you know, it's just unfortunate. Greenpeace claim to follow the science. They claim that science tells them what to do, and yet here they just ignore it. Why? Money and political power. They've gotten a lot of political power in Europe as a result of this. I think it's tragic. It's really tragic.
Rich, I've got one question. It's very exciting with all these new discovery on methylase and methylation pattern. Have you actually looked at now or how far are we from predicting, if you look at the sequence, the newly discovered method, the DNA sequence, protein structure, how far we can predict, or if we can predict the methylase pattern?
You mean, can you predict the recognition sequences?
Yeah.
The answer is not very well at the moment.
So what do you think is most difficult hurdle in those kind of predictions?
I think part of the problem is that there are many ways that a protein can fold and recognize the same sequence. And we see this particularly in the type 1 systems with these target recognition domains, 100, 150 long. We have many examples where very different sequences are recognizing exactly the same pattern. And you can talk to people like David Baker, who is a very good structural biologist who can predict all sorts of things. He's not been able to predict what's going on here. No one has been able to predict yet what's going on. So there is some subtlety there. Part of the problem, we don't have structures for most of them.
Yeah. Thank you. Any more questions from the audience? Oh, yeah. We've got actually at least 2 more.
Okay, thank you so much for your wonderful talk. Actually, I have no question. I just have suggestions. Maybe you can change this link to a QR code so we can get more support.
QR code.
Okay. We've got another question from the student.
Thank you very much for the talk. A short technical question. So regarding finding the methylation sites with the Bacterial sequencing technology, do we need to generate a negative control of unmethylated DNA every time? Or is there some baseline signature you can use as the control?
No, you really don't need to, because the Bacterial sequencing tells you exactly where the methylated base is. It is very specific. For N6-methyl-A, you get a very clean signal. N4-methyl-C, you get about an 80% signal. And it's very clear. We have thousands of examples of these now. So, statistically, we're looking really good at this. When this was first done, yeah, people were doing non-methylated DNA and comparing it with, but that was 5-6 years ago now. You don't need to do that anymore.
So does it imply that if we can hands-on some raw sequencing results, that is before base calling from the peptide sequence, we can do methylation on any data set?
Yeah.
Okay, thank you very much.
You say it's the polymerase. 529, I forgot. So now you can actually tweak the polymerase to recognize the methylation. Is that what you...
No, so what happened was that they made mutants in the polymerase. There are actually 50 mutations were introduced into 529, not to detect methylation, but so that they got an equal signal at each base.
Yes,
so that
but when they come to the methylated base, they have trouble.
Yeah.
So when you...
It was just a chance observation on their part. They had no interest in
so
this.
Therefore, you can do a systematic study by modifying P29 and to recognize and then to see which base to recognize what base.
Yeah, you could do that, but it's not necessary. Why would you do that now?
Well, but your work.
But we've got a good method for N6A, we've got a good method for N4C, now we've got a good method for 5-methyl-C.
Do you look at the sequence?
Pardon?
Did you look at the sequence or structure of the P29 DNA polymerase?
No, because the mutations actually have not been disclosed.
So my question is, why does losing the methylase make a bacteria pathogenic?
Say that again.
Why does losing the methylation enzyme make the bacteria pathogenic? You mentioned.
Oh, so the one I'm talking about is enzymes, a series of enzymes that recognize methylated DNA. So they're specific for stopping methylated DNA coming in. And most of these genes that produce toxins and produce various things that you would prefer not to have around come from bacteria in which the DNA is methylated. And so as soon as you stop, as soon as you no longer block methylated DNA, it becomes easy for genes to transfer into this strain. And that's what we saw with the E. coli strain. There's about 5 new restriction systems that popped in, and they're all in islands that are typically connected with pathogenicity. Sugar toxin is one of those that is in there. So it looks as though these methylated, methyl-recognizing restriction enzymes are really a major block to stop the transfer of extra chromosomal DNA or chromosomal DNA from getting into these strains.
Okay, thank you.
Okay, got students, and then a student over there.
Thank you for your wonderful talk. And as I know, there is a DNA methylation pathway called RNA-dependent DNA methylation in plant. So does it happen in bacteria?
Not that we know of.
So you mean RNA? Not so far. We don't think RNA is involved in bacteria.
Not that we know about. There is no evidence for that so far.
So
doesn't mean it doesn't happen. It just means no one has found it. But I think several groups have looked for it. The ones who found the systems in lower eukaryotes looked at it, as far as I know, but they didn't find anything.
Oh, thank you.
Okay.
Have you ever tried to look at methylase that work on different types of DNA? So far, I think that the methylase are working on double helix DNA. How about some other kinds like cDNA or 4 helix DNA?
Yeah, so some of the enzymes work quite well on single-stranded DNA. In fact, this methylase that is plasmid encoded works best on single-stranded DNA. There are some of the other methylases that work reasonably well on single-stranded DNA, but not terrifically well. And some work on DNA-RNA hybrids. We had a paper in nucleic acids research on that some time ago. We looked through all the enzymes that NEB sells to see which ones would work on DNA, RNA hybrids. So in general, the general rule is they work best on double-stranded DNA. Some have a little bit of activity on single-stranded DNA, but this plasmid-encoded methylase works best on single-stranded DNA.
Thank you. I guess you're the second time. Okay. Okay. No, go on. Sorry. It's a student. Okay. Maybe it's just the last round of questions. Okay.
Sorry for another question. You just mentioned that the methylated DNA actually kind of prevents the outside DNA from transferring right into the bacteria. I wonder, is there any study that whether the methylated DNA or DNA coding in the methylase genes are those regions more or less enriched with structural variation? Like, is there any genome structure study on the relevant field?
I think the short answer is not really. No one's really gone and done anything in depth on that. But just anecdotally, looking at it, that there really isn't any general rule that you seem able to draw. The specificity, so in the methylases have a region that's responsible for specificity, that tends to be quite variable.
So, so we've been a very fruitful discussion. Let's have another big round of applause to thank Sir Richard.